morning, everybody. My name is Lucien Benheim. I was chairing the work package 2. I'm from Laser Analytica. This is the small and medium enterprise that was mentioned and participated in this IMI program. Let me thank first the IMI consortium for having us into this program and giving us this opportunity to develop what I think is a very important contribution to the field. I don't know how it works. So I will be just making the introduction of the objectives of this task force. And Christine from Sanofi, and who I don't know if everybody knows, actually was the main force behind the Get-Rail program at the beginning in the industry when he was with GSK. And we are very lucky to have him come to Work Package 2 and help us after he moved to Sanofi. So the objectives that we had to tackle in the Work Package 2 was really what the so-called gap between efficacy and effectiveness. We were a methodological task force who really worked mainly on methods and as much as possible quantitative methods. And we have tried to develop tools for this. We also have worked conceptually because, as someone said, there is nothing as practical as a good theory. So if you want to develop tools and quantitative tools, you must first have a very good model to which your tools will apply. So our main objectives were to better understand when and why, and I should say maybe if, there is a gap between efficacy and effectiveness, and provide tools to address it during the drug development process. To do that, the general question, as I said, was, is there an expected gap, or so-called gap, between trial outcomes, that is so-called efficacy, and results active, or that we want to achieve in actual clinical practice, that is the so-called effectiveness. One of the problems with effectiveness, it is that most people believe that effectiveness is some sort of downgraded efficacy, a lower efficacy, something worse than efficacy, which we believe is the wrong idea. Of course, effectiveness in the real life could be lesser than the efficacy measured in trials, but actually effectiveness in the real life could be better than the efficacy measured in trials. There are many reasons for that, and one of these could be that the fact that people in the real life are not randomly assigned to drugs, as opposed to randomized clinical trials. That is the best, the people who are most likely to respond to a drug are prescribed that drug. So in the real life, you may actually sometimes anticipate that the effect of the drug will be better than what you have measured during the trial in the population. So it's a much more complex question that people would think, and we've been working on this in many respects. So the main question is, how do we anticipate it? What are the differences between trial settings and real-world practices settings that will actually drive effectiveness? So this word drive is also important. We have proposed the concept of drivers of effectiveness. And this concept is what that says is more than a word. As I said, it just said first that effectiveness is not only a downgrade of efficacy, and there are some drivers to it. So, and your goal as a pharmaceutical company, as regulators, or as decision makers, is to be able to identify the best conditions in which the drug will have the best expected effectiveness, which we call the drivers of effectiveness. And these drivers have to be measured, and they have to be modeled, and they have to be predicted quantitatively as much as possible. The second question is which studies can inform the assessment of effectiveness before launch, and that was, we were asked to try and respond to these questions. And specifically, the question is, can pre-registration trials address the populations, the comparators, the endpoints that would better inform effectiveness, or the real-world effectiveness, and how to conduct pragmatic clinical trials, but also randomized pivotal clinical trials. We work on both to tackle specific statistical issues. The question basically was here, is it possible to do a randomized pivotal study that will incorporate all the information some of the information that is needed to predict effectiveness without increasing the cost and without increasing the size and without jeopardizing the statistical power of the study. So we work specifically on these questions, and we have provided during the program, we have provided some tools for these trials to be used for these trials. So the conceptual framework is the one that we had. Is there a pointer here? Or does it point? I'm sure I'm going to do something wrong. No, that's okay. So the conceptual framework, I'm sorry that maybe you cannot see that. This is the first slide that we presented because this is the laser conceptual framework on how to go from efficacy to effectiveness and how to bridge from efficacy to effectiveness. And this is where we started with the program. It's been published, and there are some references to this. Clémentine in the room can give it to you. And the whole idea is that to be able to predict effectiveness, you must measure quantitatively, have some idea quantitatively on the interaction of the different populations with the efficacy, and some information about the actual use, how it will interact with efficacy, and how actual use will be determined by the health system as well as the population exposed. All of this can be modeled. All of this can be put into a bridging model, and a lot of the information for this can be found in existing data or in data that you can generate easily before you launch your study or your trial. So we developed this conceptual framework further within the task force, the Work Package 2 task force, to be able to provide you with the right tools for that. And Chris will give you some detailed examples of that. And then we also developed, as much as possible, the trials and how to integrate information during the trials. Here, that is, how can I generate, the question is, how can I generate evidence for effectiveness? This is a toolbox, and Chris will be presenting the details of the toolbox for you.
All mic'd up.
You have your own.
I have my own. Thank you, Lucien. As Lucien said, I switched from a kind of a general role on the program into Work Package 2 just a year ago. So I personally want to thank Clémentine Norden from Laser and Robert Olivares from Sanofi, who have been on the Work Package 2 right from the beginning and have really done so much of the work, especially in the last year, pulling everything together. So as Lucien said, Work Package 2 has looked at a lot of separate methodology problems, and what we've tried to do is to pull all of those things together into the toolbox, which you will have a chance to see later in the day. And what I want to do is just to sort of give you an extended plug for what we can show you and can discuss with you at greater length in the rest of the day. We've structured the toolbox around a decision flow. You know, thinking, for example, for here, we're going to use the example of someone in pharma R&D having to go through the process of drug development, trying to understand how real-world evidence can help in drug development. So the flow of questions that a team needs to work through would start with, why should I generate evidence for effectiveness? And the question that has to be answered is, is there a compelling need to develop evidence of effectiveness more than I'm going to generate from the phase 3 program alone? And to answer that question, you have to think about the efficacy effectiveness gap, the way that we've just outlined, understand the framework of real-world drivers of effectiveness in this particular disease area. But you also have to understand the consequences of not addressing that evidence gap. For there to be a compelling need to actually do something, there has to be the scientific driver, but there also has to be a consequence of not generating that research. And so we've also been working with HAS as one of the partners in Work Package 2 to be looking at, well, what were some of the gaps in the evidence packages presented to HAS, and what were the consequences? And we've also looked, HAS has also looked at the subsequent follow-up to those for those molecules, and what happened next when there was a gap in the evidence at the beginning, what happened afterwards. So we've had to think about the consequences in terms of the ability of HTA bodies to make a fair decision in the light of uncertainty. So if you've done the scientific workup and you realize that there's a significant risk that efficacy and effectiveness will be different, there's a significant risk that HTA will then be faced with uncertainty and a decision-making process which will be more complicated. There will then be a consequence on the HTA's willingness to make a certain decision, and then there'll be a financial consequence to the company. IMI doesn't touch on the financial and commercial implications of drug development, but as a company, you have to know where you're going with drug development and what is the consequence of doing or not doing a piece of research. So it's really important to understand that is there a compelling need from the scientific and the consequences. And in the toolbox and in of the work that's done, as I said, there is the whole report from HAS on the HTA perspective on evidence packages. There were in fact 68 pre and post authorization dossiers, so this was a significant piece of work going through the back catalogue from the French authority. And a lot of the post-launch commitments were in fact around following up on the commitments to the actual compliance with the terms of use. So there'll be uncertainty about, were the patients treated the ones we thought were going to be treated? So there is a lot of the post-launch research is about tackling uncertainty with the question at the time of launch. But what we want to do is to sort of show that there are consequences and those can be anticipated. If you, in order to answer the question from the scientific perspective, not just the consequences, you have to go and look for, is there evidence of an efficacy effectiveness gap in this disease area, in previously launched drugs perhaps, and what are the drivers of effectiveness? What are the factors that are causing that gap to open up? So we need to identify the gap, assess the consequences. We need to use a variety of different methods, all of which are in the public domain, but need to be repurposed for this particular question. So reviewing HTA decisions, literature reviews, interviews with experts, analysis of available data, collecting patient insights, and seeking early scientific advice. Each of these things on their own is not novel, but the way that they can be used to address, in particular, the question of effectiveness is the core and is the challenge to to industry and to HTA bodies preparing for the scientific advice. Is, these processes can all be adapted and used to understand drivers of effectiveness. In particular, we've published work on, written the reports and preparing the publications on literature reviews, and how literature reviews can give you insights into what drivers of effectiveness might be out there. And as you can see, we have a whole series of conclusions or recommendations that we would include in our reports. And all of this material, as I said, all this material will be available for you to come and talk to us about at the appropriate break. If the answer is yes, having done all of this work and done the literature reviews and the expert interviews, taken your scientific advice, if you really believe that there is going to be a significant gap between efficacy and effectiveness, and you think you've got a good clue on what the drivers might be, the next step is to say, how can I address that? How can I address the evidence of effectiveness? What are my options in terms of study design? And remember, we're pre-launch, so we can't just do an observational study. We have to be thinking about adjusting our registration studies or commissioning phase 3B programs in a pragmatic way. Or we need to be moving into and putting more challenges on our analytical plan and our evidence synthesis plan. The work package 4 will be picking up on a lot of the analytical work that the consortium has done. But we've done a lot of work in terms of thinking on the theoretical and statistical challenges of the implications of trying to adjust your trial program, trying to bring pragmatic elements into a trial, trying to design pragmatic trials themselves. Work Package 3, you'll hear about, has done a huge amount of work on the operational, ethical, legal issues of trying to run and get pragmatic trials up and running. We focused on some of the statistical issues on powering and, for example, a very interesting piece of work on, can I enrich my trial population? Can I look at my RCT? I recognize that it's not including a population which is going to be of huge interest and importance in determining the effectiveness of a drug. For whatever good regulatory reason, patients may have been excluded completely from a trial program. How can I address that by reintroducing those patients into the trials in a way which gives me some information about effectiveness but doesn't jeopardize my clinical trial program? So I'm not looking for a label claim on a particular population. I'm just looking for some information. I'm looking for something that's better than nothing on a population of interest. And so these are the sorts of questions that we've addressed and taken through, for example, here, recommending a series of steps to how to enrich the trial population that will allow you to assess is this a feasible solution for that particular effectiveness problem. Another very interesting topic which Tjeerd van Staars and Manchester were leading on was the issue of sort of running pragmatic clinical trials, within a real-world cohort, and what are the statistical implications of completely, taking that cohort for that trial and randomizing it from a real-world population. And there's a very interesting question, I think, which you can follow up with the appropriate research group, which is saying there is a potential risk that the patients who are identified and pulled out and asked to consent to join the trial but refuse to join the trial are in some way different to the patients who consent to be in the trial and then are randomized to a usual care arm. So is there a systematic difference between usual care for patients who did not consent, usual care for patients who did consent, and the intervention arm? And I think there's some very interesting work there. But this is the kind of statistical problem that has to be anticipated in order for us to have confidence in running and then communicating the results of PCTs to our customers. So we've put this whole framework in a poster with all these links. We've put all this together. So all of this is going to be outside for you to take a longer look at. And all of the materials and reports and more detailed advice is going to be included in two ways. First of all, the toolbox itself being made available as a standalone PDF document so that you can take away and as a separate thing. But also, we've been working hard with Work Package 1 to make sure all of this decision-making flow and the tools and options is all embedded in the Navigator. So the Navigator, as Pal said, and Rob said, was a Work Package 1 deliverable on behalf of the whole consortium. So that decision-making flow and a lot more detail on what the tasks that we've been looking at in particular in Get Real will be available. In terms of how we think this should be used, we think it's both the HTA and the R&D teams can benefit from thinking about the efficacy effectiveness problem in the same way and understanding what may be reasonable approaches. So when HTA and R&D come together at scientific advice, for example, that there's a lot of common ground in terms of thinking how we think about that problem in a structured way and how we think about what reasonable trial demands might be before launch. And I think that will help. Really, we want to feed a lot of this thinking and this framework of thinking into the scientific advice initiatives that are ongoing, so that those scientific advice programs can help companies and HTA bodies not just think about the registration trials, but think about the total package of evidence that's required. We also, where we have publications already out, we've brought, Clementine has brought along some reprints, so we have some papers that can be taken away or, of course, looked up online, as is the modern way. But we certainly look forward to talking in more depth with any of you outside of this session. Thank you very much.